Good morning, students. Welcome back to lecture 5. We will talk today more about antigens. Specifically, we will spend more time on discussing antigens. So from past few classes, we have been studying antigen-antibody interaction, the basics of it. We understood that antigen is a toxin or other foreign substance. Antigen is a toxin or other foreign substance which induces an immune response in the body, especially in terms of production of the antibody specific to the antigen. An antigen is a toxin or other foreign substance which induces an immune response in the body, especially the production of antibodies. That was the basic understanding we were discussing first few classes. And also we discussed a lot in the last class about B lymphocyte in terms of immune cells, lymphocytes, types of lymphocytes, B and T cell lymphocytes, then how they elicit specific humoral and cell-mediated immune responses. We have spoken a lot about the type of specific structures present on the specific cells, how they interact with the antigens we already discussed. And also we discussed that the antigens are mainly the important fundamental aspects for that is that the self and non-self recognition. Identifying self from non-self is one of the most fundamental importance for immune response. We already spoke about it in terms of MHC molecules and other recognition patterns. Thus, by summarizing the previous two slides, we can redefine the antigen-antibody interaction as one more definition. For example, antigens are substances that can be recognized by immunoglobulin receptors of B cell or by the T cell receptor when complexed with MHC are also called as antigens. So you are not talking now in terms of an antigen inducing an antibody response in this direction. You're also talking in terms of the whole immune system specifically moving forward to recognize an antigen. It's a two-way definition, not only a toxin that elicits an immune response, or you can say in other words that substances that can be recognized by immunoglobulins, antibodies present on B or T cells, and specifically in T cells, they are complex with MHC molecules. Those are also called antigens. So coming back in looking in terms of how the antigen-antibody interaction happen in live in cells or tissues in vivo, you just look in at the different structural organization of the tissues and organs. For example, you start with certain specific cells in action. For example, how this process happens, you take certain cells in action, for example, immune cells here, and in terms of here, you're talking into specific 4 kind of cells. This cell is in the green, they are bacterial cells. They are the source for producing the antigens. And they are recognized by specific cells called as immune cells. Probably here is a mast cell or probably mast cell. Is trying to recognize the bacterial cell with the antigen. And one of the thing here is the red blood cell that is there. So a kind of immune cells, they come in action and they target some of the antigens present in the bacteria and they go endocytosis, they kill that. That we already see. So one concept you understand here is cells are in action. That is one fundamental overall organization of the interaction you see. Cells in action. Then you come into another level of organization called molecules in action. We already know that a bacteria will specifically represent a specific chemical molecule or structural molecule called antigens. And in terms of the cells of the immune cell produce either antibodies or they produce T cell receptors, through that they can communicate. So you look in terms of these molecules are there in action, working in concert or in cohort. Again, you go back into another structural organization where two kind of concepts we can discuss. Cells in action, molecules in action, but structures in action, and then chemical bonds in action. For example, you see here antibodies and antigens here. We already know that there is a lock and key model works, ligand receptor mechanism works. We already know that the antibodies will have specific grooves, structural grooves, where an antigen come and fit structurally. This structural uniformity is really important. If there is no proper structure between this and these lobes, this antigen antibody recognition will not happen. So you should remember that the structures also work in action. That is very important. And when you also looked at in the structure of antigen antibodies, specifically in the antibodies, We spoken in the last class that they have two chains, long chain and short chain. We already know that there is variable and constant region, but it should be remembered that they are nothing else. Antibodies are protein molecules, immunoglobulins. Then we will talk in few slides fundamentals of the proteins. They are just made up of bonds. The bond is mainly disulfide bonds, peptide bonds within the amino acid. So you go close into that, you are looking into chemical bonds in action. Probably a scenario of you are looking in these groove structures here. Here you are looking at that. So here this yellow is the antigen presented by antigen presenting molecule in the green. And you see here the antibodies here, red and blue, short and long chains. You see that there is a structural grooves and a specific chemical bonds that align themselves in this epitomic region we talk in the few slides. So there is a organization that comes in terms of antigen antibody interaction. You are looking at the holistic development of this interaction. You start with the cells, you go with molecules, then you go with structures of the molecules, how the structure interaction is happening, and at the end you conclude with the chemical bonds. So if you want to talk about immunology per se or any other biological concept, this small chart is enough to talk about that. It's just like you are looking at a diverse or a different biological process, different chemical molecule, different structural organization involved. But in terms of the basic understanding, this is the overall workflow that happens in the body. So please try to understand that. If you understand this concept, you can write any of the concept in immunology, taking a specific example of in each category. So coming on to this concept, you can pose some questions. So where do we get the antigens from? For example, we are talking about antigens today in the class. Where do we get the antigen? We already spoken that there may be pathogens that produce antigens. That one case. But how about self-antigens? You know that there are antigen molecules, endogenic to the molecules present in the body. They are not from foreign organisms. There are self-antigens. So not only pathogens, there are self-antigens present in the body. So one more important concept you need to understand that, one question is that, do immune cells recognize self-antigens by birth itself or foreign antigens by birth itself? They have a specific blueprint that they should know about everything by birth, or by encountering them throughout the life cycle of the immune cells. So you can ask two questions. Either they know by birth or they learn it by experience. They learn it by exposure to a specific antigen during its development. For example, an immune cell that starts from lymphopoiesis, B lymphocyte, that can go into two phases of immature cells, mature cells. So during the development, we already know that they go through lymphoid organs, they go through all over the body. So by traveling as an immature cells also, they get exposed to certain antigens. They try to remember the structure. So basically, the immune cells recognize, learn by encountering or by expertise or experience. And also in terms of what are the structural and physical properties of antigens, are they chemical molecules? If then what are these micro or molecules, macromolecular in nature? How do you say that? Or micro or and then size and chemical nature of it also important of the antigens. Also, do all antigens elicit immune response? So whatever chemistry, whatever you talk here, are they all potential enough to induce an immune response? Or if not, we will study all that in the coming class, in the coming slides. And also in terms of the dose, does only one dose is enough? Only one dose fits all? If not, how to enhance or induce an immune response? So one dose of an antigen or several doses of antigen in terms of vaccination, we know that. For example, BCG vaccination, you may give it in small doses. You have to finish all the cycles to get complete immunization for that. So you consider all these questions. So that will be the essence of this lecture today. I will cover the lecture today into the following concepts. For example, we'll briefly talk about immunogenicity and antigenicity, basic structural, basic functional aspects of proteins. What are the basic units proteins made up of? What are the other macromolecules? What is protein-protein interaction, protein-non-protein interaction, and antigens, adjuvants, and haptens. We will talk in this class. Please reminded that the class is a bit of descriptive because we need to introduce a lot of definitions, a lot of new terms. Majority the class goes in a descriptive fashion. Once the lecture is over, we can have a discussion or a overall brainstorming session, or we can have a question section. Separate class can be done for that. If you don't understand a concept, we can take it further. We can discuss an individual concept again in more details. So just imagine our human body. So you're looking in a human body, a cartoon diagrammatic. Our body is very complex, right? It has complex in terms of chemical, highly complex and diverse chemicals are there in our body. For example, human body, the complex organization of chemical molecules and their interactions and their pathways. In summary, you can take that we already know that central dogma of molecular biology, how a gene codes into a specific protein and it goes through a process of RNA production, transcription, translation. And immunology, most of the immunology you are talking about is specifically into focusing on these proteins, because most of the immunoglobulins or most of the molecule pathways concerned here, they exist only in this protein forms. But in minority, you can see that there is a nucleic acid, there is some lipids, some other nucleotides can also act as antigens. That we will look into the further slides. But try to remember that human body is complex. We have basic fundamental units. We have metabolic units, micro and macromolecules that are building blocks of different structures and proteins. So as I said, proteins. We need to talk a bit more about fundamentals of the protein to better understand further classes what we are talking about. So we all know that proteins are made up of amino acids. Polypeptide chain, monopeptide chain. We know that amino acid 1, amino acid 2, amino acid 3. It is a a kind of protein made up of 3 amino acids. It's a monomer or not, it's not a monomer, it is a polymer of 3 amino acids, a polypeptide chain. We already know that the amino acids consist of N-terminal and C-terminal, and we know that the peptide bonds, that removal of the hydroxyl group, water molecule here, causes a peptide bond between amino acid 1 and amino acid 2. Like that, it goes through the next cycle and a polypeptide chain will form. Different amino acids are coming up here. And then this is a primary structure, the coding of specific amino acids specific to a protein. A specific number of amino acids will be there. They form a polypeptide chain. That is the primary structure of the amino acids of the protein. Then it goes for a secondary structural organization. For example, there is chemical interaction, chemical bonding happens within this amino acid. There will be a kind of sheets and helices will form in the secondary structure. Again, this organization goes into another conformation change called tertiary structure, where there will be three-dimensional organization. And again, one protein on this three-dimensional has to interact with several complex of proteins to elicit or to enhance its functional interaction or do its function properly. So you are looking for a 4 protein molecules in a complex here. So try to remember that the protein structure, organization, conformation is really important for its function. So you are looking for an example here, starting from again nucleic acid, DNA. You are looking at the protein code. For example, of the insulin gene here, mRNA sequence is here and the coding sequence, and the amino acid sequence is here. These are the letters that represents amino acid or specific length of the insulin. So we already know that we have 20 amino acids coming from 64 codons. I don't want to spend time on that. It's a fundamental you should be knowing about the molecular biology. So we have 20 amino acids. I just want to emphasize here on the chemical structure of amino acids. You can go here, the chart represents different amino acids. It's a kind of chemical structure, code of the letter, and then whether it is an aliphatic, aromatic, you can see a specific color code of the amino acid, aliphatic or aromatic, either is it acidic or basic, and one more important, it hydroxylic, either in terms of hydrophilic or hydrophobic, or specific compounds of sulfur amides present, non-essential and essential. It may not be necessary for this class. Try to go through this slide in your free time. Try to understand in terms of the chemical nature of amino acid. This imparts the tertiary and primary secondary conformation of the amino acid consistence of 20 or 30 polypeptide chains. You know that there is different conformation structures present. They can interact in a heterogeneous way. That is really important for a protein conformation. Once a protein is formed, there is another concept called post-translational modifications. A protein that is already formed structurally in the tertiary structure conformation or primary to tertiary, or in terms of quaternary structure also, the protein amino acid molecule, the basic units, they may link by disulfide bonds. They may have structural or chemical moieties that can be modified again further. That is called post-translational modifications. A modification can be either of chemical groups, methylase, phosphorylase, and acetyl groups can attach to specific residues here, or it may be specific protein molecule, ubiquitin or sumo molecule can attach to the molecule, or some complex molecules of sugar. For example, glycosylation is a process where these molecules of sugar moiety will attach. And the fundamental concept is that again you can break the disulfide bonds to study individually. That is called proteolysis. Try to understand this concept because that is really important for antigen-antibody interaction, because the antigen may be existing in one of these forms that we will see in the next slide. So if the antigen is a protein molecule, it exists as a proteins, it may exist as one of these forms. It may be presented to the antibody either as a primary structure, secondary, tertiary, or quaternary structure. Specifically, an antibody attaching to this protein molecule, an antigen present in this complex, may interact from this groove. It may interact from an active site here in this direction. But the protein may still exist in a complex with other molecules in the other sites. But antigen-antibody interaction can happen here. It may happen with the alpha helix. It may happen with the beta helix. It may happen in one of the groups here. It may happen with a specific phenylalanine or glycine residue here. To try to understand that, the structural conformation, presentation of the antigen is really important. It can be any one of the form. And majority of immunogens are proteins in nature. We will see why you are using the term immunogen in the next slide. There are majority of them are proteins in nature. So let's take some time and just glance through the text here in this slide. This slide focuses on two concepts. One is immunogenicity, one is antigenicity. Okay, try to remember the terms. Try to look here. Antigens can be any of these form. So look in the two terms, immunogenicity and antigenicity. So we already know that immune cells are here. The antigens are chemical molecules that may come across here. That's what we discussed in the fundamental subtype. So just look in the fundamental aspect. By mere attachment of antigen to either T cell receptor or the antibody in the immune cell may not elicit an immune response, because we are talking about chemical structure that present on the cells. Chemistry and protein-protein interaction can happen here. That is called as antigenicity. A chemical protein of antigen a chemical molecule of antigen can interact with the molecule present in the immune cells. It's just an antigenicity property at the fundamental level. So a kind of chemical interaction you are talking. But whether it's gonna elicit an immune response, this interaction's gonna make a B cell to go into plasma cell and memory cell, produce a pool of antibodies, that is not yet we don't know unless we see these steps going through the completion and immune response. So basically, immunogenicity is the ability to induce humoral and cell mediated immune response that comes through antigen antibody interaction. If a case of antigen antibody interaction, they can elicit the immune response through this completing all steps, that is called immunogenicity, and that molecule is called immunogen. But by mere attaching to these cells and it doesn't go to the completion of any response, that molecules are called antigens and they have antigenicity and they don't have immunogenicity. So basically look to the abbreviation here. Immunogenicity is the immune response. Although a substance that induces specific immune response is usually called antigen, and unless it does the immune response, it is called immunogen. If not, it is only called as antigen. Antigenicity is the ability to combine specifically with the final product of these cells, but it may or it may not elicit an immune response. The fundamental differences that leads to us to conclude the fact that all immunogens are antigens, because whatever that causes immunity or immune response are all antigens. But at the same time, the converse, all antigens are not immunogens. Please try to understand the fact. It is really important fundamental concept. So how an antigen become an immunogen and it can elicit or have an immunogenicity. That's one more fundamental. You can ask a question there. To do that, to elicit an immune response, a specific antigen molecule should have some of these salient features. For example, foreignness of the molecule. How foreign is it compared to the body of a specific organism or specific endogenous pool of molecules present? How foreign is it? That is really important. And also molecular size of the antigen is also important. And chemical composition and heterogeneity. We already know that most of the antigens are also proteins. What are the proteins made up of? How heterogeneous are the proteins? They have only phenylalanines of all the 40 amino acid that code for the protein, or they have a mix of phenylalanine, valine, some other, or they may have a mix of aliphatic and aromatic. They have a mix of hydrophilic and hydrophilic amino acid. That is also important. And susceptibility to antigen processing and presentation. This concept we already discussed well in the last class when there is a antigen presenting molecule present the antigens to T cell receptors, T helper cells. That we will see again briefly. So coming on to foreignness. So let's see here. Foreignness means a self molecule. Let's say an example. The system, the immune system or itself, the body is tolerant to a self molecule. Let's say snake venom, it produces toxins, but snake will have this toxin roaming all around its body, but it is not toxic to snake because it is a self molecule. The immune system has adapted to gain tolerance to the snake venom, so they don't kill the snake by thinking that this is a non-self molecule. But coming on to any non-self molecule, it may exist in the form of intolerant molecule. The immune system is not tolerant to that. That is the fundamental difference. And also we, as I explained in the previous slides, that during maturity of lymphocyte, from immature to mature cells traveling through all of the body, it might be exposed to all the self and non-self, and it may gain a tolerance or intolerance to that. That one more important aspect. And degree of immunogenicity is proportional to degree of foreignness. For example, the more foreign is the molecule in your body, the higher the immunogenicity or the higher the immune response. That is one more important concept. And at the same time, how do you say a foreign molecule? From humans, rabbits, mice, insects. So you have to look into the phylogenetic tree and the phylogenetic distance. We evolved from single molecules, single organisms to complex organisms through phylogeny. You can through evolution. The more the phylogenetic distance, the more degree of foreignness, the higher the immunogenicity. That is also one more concept you need to understand. For example, bovine serum albumin, BSA, one of the molecule working as an antigen, is non-immunogenic to cow because bovine is coming from cows. It is non-immunogenic to cow because same species coming from same animal. But it is immunogenic to rabbit. You see that? Why we use in terms of vaccination also? Why use a different species, different organism? Because you want to elicit the immune response to the maximum. So BSA produced is non-immunogenic to cow, but it is immunogenic to rabbit. And also chemical composition and heterogeneity. For example, you are looking here specific molecules of antigens that are here. You see bovine gamma albumin, bovine gamma globulin, BCG, bovine serum albumin, BSA, flagellin, ovalbumin, tetanus toxin, TT. Try to understand basics of our vaccination. These are the molecules or compounds used for vaccination also, right? Vaccine is an antigen, maybe living, non-living molecule. We will talk in the vaccination chapter more about chapter 7. But try to understand the fundamental nature of these antigens. Mainly they are globulins, means they are proteins in nature. And you see some other molecule that may be a lysozyme, enzyme, hemocyanin. And look in the size of the molecular mass here. In terms of BGG is 1,50,000 Daltons. And one more KLH is greater than 2 million Daltons. And the more the size, the more complex, the more important is the immune response. For example, synthetic proteins with same amino acid residue fail to elicit immune response. There is no heterogeneity. You make an artificial protein having only valines, having only phenylalanines. the homopolymer. You try to induce through that antigen and immune response, you may not succeed because heterogeneity or heteropolymerase is a fundamental aspect to raise an immune response. And also higher the complexity, higher the size of the molecules, in terms there is more competitiveness in induced immune response. The higher the size, higher the immune response. Also lipid molecules sometimes used as antigen, that we already, some of the lipid molecules can be used as mainly proteins, and lipid protein complexes also can exist as antigens. So fundamentals of antigen nature, size of the molecules, and also chemical composition. For example, in terms of lipids here, we have two different lipid molecules, leukotriene C4, prostaglandin D2. They are also worked as antigen, but look in the complexity of the structure of the lipids here. So if it may look complex to you, but if you compare with the lipid molecules, it is look like simplistic molecule. Compared to leukotriene, the complex structure and nature and the bonds here, you take a scale of 1 to 100, how this antigen can elicit immune response, how this antigen prone to immune response. You see that higher the complexity of the lipids, the immune response is 100%, 100 the value. But prostaglandin is very less immune response. Okay. So coming on to susceptibility of antigen processing and presentation. Already we discussed in the last class all the steps involved. I would like to again emphasize on the fundamentals here. You go through and read the steps here again one more time. So some endogenous antigens are already present. Coming from bacteria, they might be processed here. So you are talking about two antigens, two pathways. This is endocytic pathway. For example, exogenous antigens coming from bacteria, they go through endocytic pathway, and cytosolic pathway where antigens already going through the body fluids, they can be taken to cytosolic pathway, endogenous antigens. Processing refers to degradation of proteins into peptides by proteolysis. What is processing is that proteolysis can happen, proteins can be degraded into peptides, and presenting refers to binding of peptides to MHC molecules and later in the complex to T cell receptor. So an antigen having a specific some chemical, either lipid protein molecule, should be able to undergo all these changes properly, should be able to go into a final form that is required here, and should be able to represent it to the T cell in a proper. That is also an important concept required for that. So how do you give a dose of antigen or immunogen? Dosage and route of administration is also important. You can go all these methods that we already know fundamentally: intravenous, intradermal, subcutaneous, intramuscular, intraperitoneal. Why there are different mechanisms? Why not through the digestive tract? Why don't we take orally the vaccines? Because they might have a specific location they want to reach and elicit a specific response in the body. For example, intravenous, that may an antigen that is given intravenously, it just quickly goes and first reaches the spleen first. And we know that in the spleen, what are the immune lymphoid organs present, what are the immune system present. And again, subcutaneous, it goes to the lymph nodes, specifically targeting a specific immune cell. That's why the route defines which organs and cell populations the immune system will be involved. That is an important for administration. So a few more slides. I might have to go descriptively here. So go and look in your free time, read the abbreviations, read the definitions more keenly, make notes of it. One more concept we are talking here, isoantigens or alloantigens. They are synonymous, both are synonymous. An antigen derived, for example, isoantigen, antigen derived from one member of a species that can cause the production of antibodies in some other member of the same species. For example, myself and some of the students in this class, you might have a specific antigens. I can take that derivation of the antigens from you through antigen antibody interaction, make them specifically take the antigen from your body, make it chemically, then put it into my bodies, because I may not have the antigen present in your body. It may be specific to your body, specific to your one member of the species. So isoantigen present in one member of species taken into another member to elicit specific antibodies. You can see that it may be red blood cell antigens, histocompatibility antigens. You can go through that. You can read more on the iso and allo antigens here. Coming on to another concept called adjuvants. In immunology, adjuvant is also an important concept used. An adjuvant is a molecule derived from Latin. Adjuvare means to help. Again, it's try to help the immune system, try to help the antigen. Adjuvants are substances that, when mixed with an antigen and injected with it, enhance the immunogenicity of the antigen. We know that antigens, all antigens may not be immunogenic, but to enhance their immunogenicity, you can use a specific molecules in a combination of cocktails. They can boost the immune response. How they do that? It's a molecular biochemical mechanism is not completely understood, but there is a specific postulated mode of action. For example, an adjuvant combined with an antigen prolongs antigen persistence in the body, enhances a co-stimulatory signal where antigen is represented to the molecules of the immune cells, T cell, in a good manner, in a pathway specific manner, in a co-stimulatory signal manner. Can induce granuloma formation, can stimulate lymphocytes non-specifically also. If maybe there is not much of specific interaction, it may elicit a non-specific interaction. Look in the specific different adjuvants. For example, alum is used here. They can do specific responses. Each molecule will have a preference to one of the pathway explained or one of the mode of action explained here. Okay, we are running close to 5 minutes. Hopefully, I will try to cover max. Otherwise, we will speak in the next class. And also look in epitopes. So we know that antigen antibodies are protein molecules, let's say, but they are not interacting as a molecules as a whole. They are interacting at a specific active sites. Those sites are called antigenic sites or epitopes, antigenic determinants. The epitope of the protein may be in the primary, secondary, tertiary, quaternary structure that we already discussed. It may be a polysaccharide, it may be a branched sugar that may also specific conformation. And the B cells recognize mainly soluble antigen. B cells are humoral, right? They exist in the body fluids. They mainly recognize soluble molecules that are membrane-bound antibodies, a soluble antigen. That is important for that. How it is exposed, how it is soluble in the body fluids, how is represented to that. And T cell mainly recognizes peptides along with MHC molecule. So again, a fundamental distinction can be made between the epitopes, between B cell and T cell, how they are represented. You go in the characteristic here, you can see the fundamental differences here. Chemical nature, protein, polysaccharide, lipid that kind of exposed to B cells, and T cells, mostly proteins. You can go into the organization here. Again, you go into the terms of active sites and bonds. B cell epitopes are structurally soluble, structurally conducible to be represented to the antibodies here, antigen here. One more fundamental concept. You can go through this slide. You can easily understand what I explained in the previous slides. For example, T cell also, one more concept. There is a ternary complex that forms. This is a T helper cell, membrane of T helper cell, membrane of antigen presenting cell, a class 2 MHC molecule associated with the antigen here. Specific T cell receptor will be there. Again, T helper cell has CD4 positive molecular signature. So to have a proper epitope, antigenic interaction of this epitope here attaching to helper cell, you need to have a ternary complex forming index with MSC. That is fundamentally important for that. Coming on to another molecules called haptens. These are also specific to haptens are also specific to adjuvants, but it's derived from Greek. It's called to hasten the immune response. It is a small molecule. It may be combined with a large carrier molecule such as protein. We will see in the next slide. It is basically an antigen. It may combine with a specific protein molecule or protein structure. It may be immunogenic. Many drugs like penicillins are haptens. Then a hapten is actually incomplete antigen. Most common haptens are globulins, albumins, and many other. They are antigenic but not immunogenic. To enhance their immunogenicity, you need to have a carrier protein molecule, hapten carrier conjugate. These are the hapten molecule in a synthetic molecule of a conjugate made here. A hapten combined with a carrier immunized in the rabbit. They will have specific enhancement here. Specific hapten go on and attach the antibody. It may not elicit an immune response, but a combination of these three various molecule, diverse molecule coming from this can elicit the immune response in a better way, not going alone in terms of hapten. That is the hapten carrier conjugate here. You can look into that in more detail. Synthetic polypeptides such as polyl-glutamic acid, polysaccharides, and liposome molecules can also be used as a carrier for hapten here. So fundamentally, haptens and adjuvants differ in terms of complexity of the molecules, in terms of pharmacological importance here. They are more likely chemical in nature here. They are more likely pharmacological. When conjugated, a response will come. So basically, that comes to the conclusion of the class. It's just fundamentals of antigens, a lot of descriptive text you go through in free time. Try to go through these 2 slides that explains one of the company try to produce antibodies for COVID-19. They are trying to find specific membrane proteins, S and N proteins, try to raise the antibodies for that. Go through these 2 slides. Okay. So next class, we will talk about antibodies, genetic basis of antibody production, diversity, how heterogeneity of antibodies will come up. We will talk on Friday. I will post this recording. Okay.